Okay, so what if we could turn any cell in your body into any other type of cell, completely on demand? I mean, think about it. Skin cell to heart cell, just like that. That's the incredible promise of cellular reprogramming. But for years, let's be honest, it's been more of a messy art than a precise science. So, in this explainer, we're going to break down the powerful genomic tools that are finally turning that art into a true engineering discipline.
Here's how we're going to tackle this. First, we'll look at the fundamental puzzle of reprogramming itself. Then we'll dive into the single-cell revolution that really let us see the problem clearly for the first time. After that, we'll get into how we can decode the cell's decision-making logic, trace its journey over time, and finally, we'll look at the mind-blowing ways scientists are learning to record the entire blueprint for success.
You know, cellular reprogramming really holds the key to a medical revolution. Seriously, imagine generating brand new heart cells to repair damage after a heart attack, or creating patient-specific neurons to study Alzheimer's in a dish. This isn't science fiction anymore. It's the very real, tangible promise of this field.
And that optimism? It's totally well-founded. Discoveries like the Yamanaka factors, this specific cocktail of proteins that can literally rewind an adult cell back to an embryonic-like state. I mean, that just blew the doors wide open for regenerative medicine. The problem is, turning that amazing breakthrough into a reliable therapy has been blocked by some really persistent technical hurdles.
So here's the core of the problem. For all its incredible promise, the whole process is just wildly inefficient and unreliable. The cells we manage to create, they're often developmentally immature, kind of like half-baked. They lack fidelity, which means they might turn into the wrong cell type entirely. And the final product you get is usually this impure mix of successes and failures. These three barriers right here are what have stalled real-world applications for so long.
Right, so to solve these problems, scientists first needed to actually see them clearly. For years, they analyzed cells in bulk, and the best analogy for this is trying to understand a fruit salad by chucking it all into a blender and making a smoothie. You get the average flavor, sure, but you'll lose all the crucial information about the individual fruits.
And this slide just perfectly illustrates that massive shift in thinking. On the left, you've got that blurry smoothie of bulk analysis. But on the right, single-cell analysis gives us this stunning, high-definition snapshot of every single individual cell. And suddenly, those incredibly rare, successful cells are no longer hidden in the average. We can finally see the true, messy, complicated reality of the entire process.
So, how does this amazing technology actually work? Well, it's a pretty clever four-step process. First, you break up the tissue to get individual cells. Then, you encapsulate each one in a tiny droplet with a bead that has a unique barcode on it. This barcode tags all the genetic material from that one cell. After that, these tagged libraries are prepared and sequenced. And finally, powerful computational tools put all the data back together, giving you a super detailed molecular profile for every single cell you started with.
And the results were just wow. Single-cell RNA sequencing didn't just show us a simple mix of start cells and end cells. No, it revealed this whole complex ecosystem of different outcomes. We could finally identify and categorize specific off-target fates, cells that accidentally became neurons instead of heart cells, and these so-called dead-end states, where cells just get stuck in some kind of intermediate limbo, totally unable to finish the journey.
Okay, so knowing what kinds of cells are in the final mix is a huge step forward. But the real question, the one we really need to answer, is why? Why does one cell choose one path over another? And to figure that out, we need to look deeper than just the genes being expressed. We have to examine the cell's underlying regulatory logic.
To get to that why, scientists use a technique called scATAC-Seq. Now think of the genome as the cell's complete instruction manual, this massive book. Well, scATAC-Seq basically tells you which pages of that manual are currently open and being read in each cell. It reveals the active control panel by mapping which regulatory parts, like enhancers, are physically accessible for proteins to get to.
And this really gets to a core reason for why reprogramming fails so often. The factors we add to the cells are like a key, but what scATAC-Seq showed us is that the lock, the target gene, is often tangled up in what we call epigenetic bottlenecks. The DNA is just packed so tightly, it's completely inaccessible, so the key simply can't get in to do its job.
Now, this is where it gets really cool. By combining the what we learned from scRNA-Seq with the where from scATAC-Seq, we can start to build a model of the how.
This is done through something called gene regulatory network, or GRN inference. You can think of it like reverse engineering the cell's entire operating system, or maybe drawing out its complete circuit diagram, mapping exactly which switch controls which light.
And the power of these GRN models is just immense. They let us move from just observing the system to actually predicting how it will behave. Scientists can run experiments right on a computer, in silico, simulating what happens when a factor is added or removed. This lets them design much smarter cocktails of factors to get past those bottlenecks we saw earlier, and it saves months, if not years, of painful trial and error in the lab.
So we can see the start points and the end points with incredible resolution now. But programming isn't a static picture, right? It's a dynamic journey. So the next major challenge is to actually track these cells over time to really link a cell's initial state to where it ultimately ends up.
So scientists have two main ways of doing this. On the left, there's trajectory inference. This is a computational prediction. It's sort of like trying to map a car's journey just by looking at a few snapshots taken along the highway. But on the right, we have lineage tracing, and that is the experimental ground truth. This is like putting a unique genetic GPS tracker on each and every cell, so you can see exactly where it and all of its kids ended up.
And lineage tracing led to this really profound discovery. It turns out that reprogramming isn't some completely random lottery. There are actually these rare reprogramming permissive cells in the starting population that are, for whatever reason, intrinsically primed for success. This means the cell's origin has a massive influence on its final destination. It changes the problem from one of pure chance to one of identification and selection.
So this brings us right to the cutting edge, to the future of the field. If we know that these specific successful journeys exist, how can we possibly capture every single twist and turn along that path? How do we build the ultimate, step-by-step instruction manual for engineering a perfect cell?
Well, the answer might lie in this futuristic-sounding concept called molecular recording. These are brand new technologies that literally turn the cell's own genome into a hard drive, a kind of molecular tape recorder that creates a permanent, readable log of key events as they happen, in real time. And here are two incredible examples. There's DNA typewriter, which uses CRISPR gene editing to act like, well, a typewriter, writing a history of events into the DNA in the exact order they happened. Then you have ENGRAM, which is even more advanced. It doesn't just record that a signal happened, it records its identity, how strong it was, and for how long, all written onto the very same strip of DNA. It's just amazing.
So the ultimate goal here is to use these molecular recorders on those rare, successful cells that we were able to identify. If we can do that, we could capture the complete blueprint for success. It would be like having a perfect film of the entire journey, allowing us to read out the exact sequence, the timing, and the dosage of every single event required to perfectly engineer any cell we want.
You see, all these methodological advances are giving us this absolutely unprecedented view into the machinery of the cell, and they leave us with a really tantalizing question. With these tools in hand, are we on the verge of moving beyond the random, stochastic art of cellular reprogramming and into a brand new era of deterministic, predictable cell engineering by design?